Hello everyone, my name is Chris Childers. I'm from the Virginia Tech Carilion School of Medicine, and I'm presenting the project Focused Ultrasound Biofilm Ablation, Investigation of Histotripsy for Treatment of Urinary Catheter Biofilms. Our clinical problem of interest is treating urinary catheter biofilms. Specifically, we chose this problem because among UTIs acquired in the hospital, approximately 75% are associated with urinary catheters. Between 15 and 25% of hospitalized patients will also receive a urinary catheter during their hospital stay, and biofilms are readily developed on the inner surfaces of these urinary catheters, leading to clinical infections. Current treatments include antimicrobial catheters, surface modifications to catheters, and urease inhibitors, but all these face significant limitations such as antibiotic resistance, limited efficacy, and use of systemic drugs. Histotripsy is a non-thermal, non-invasive ablation method that uses high-pressure ultrasound pulses to ablate tissue with high precision. In the top left here, you will see histotripsy treating within the liver, and in the top right here, you will see histotripsy being used to ablate red blood cells and a red blood cell tissue phantom. Histotripsy has also been used to disrupt bacterial biofilms on glass microscope slides, graphite discs, and surgical meshes. A histotripsy lab at the University of Michigan has also used histotripsy for thrombolysis, restoring blood flow to a vessel phantom model. The goals of this project is to determine if well-confined histotripsy bubble clouds can be generated inside urinary catheters, and also to demonstrate the feasibility of using histotripsy for the removal of luminal biofilms, and finally to characterize the bactericidal effects of histotripsy on suspended bacteria inside catheters. Here in Figure A on the left, you can see a schematic of our treatment system. It includes a 3D positioning system, histotripsy treatment transducer, 90-degree high-speed optical camera, and coaxially aligned ultrasound imaging transducer. Figure B on the right is a real picture of our transducer system, and we use this system to confirm the feasibility of luminal catheter cavitation. The hypothesis that we tested with this first experiment was that histotripsy can produce cavitation within the lumen of different materials and sizes of tubing, as well as clinically used catheters. The results of this experiment showed that well-defined bubble clouds were generated inside the lumens of all catheter types without inducing prefocal cavitation. Here, the materials tested were Tygon, silicone, and latex tubing in various inner diameters of 1/4, 1/8, and 1/16 inch, as well as two brands of clinically used urinary catheters with the brand and model included in the lower right corner. These data show the well-defined bubble clouds in the longitudinal view using a 90-degree high-speed optical camera as opposed to previous cross-sectional ultrasound imaging. These are still images from the previous videos showing clear luminal cavitation. The purpose of this data is to demonstrate the geometric similarity of our Tygon catheter model, figured here as Figure C, and compared to our clinical catheters, Figures A and B. We used Tygon catheter model in our experiments due to its superior imaging characteristics the Tygon catheter model provides for these pilot studies. In this next experiment, we used our treatment system as previously described to treat a cultured luminal biofilm. On the right, you will see a validation of the cultured biofilm within the lumen of our Tygon catheter model, along with the corresponding spectrophotometry data after solubilizing the crystal violet stain. We then treated this biofilm by treating a pre-designated 15.5 millimeter treatment zone with 200 pulses per step and a step size of 0.1 millimeters. Here is a video of one of our treatments in process. You can clearly see the optical as well as cross-sectional ultrasound imaging, and you can see that portions of the loosely adhered biofilm were removed rapidly by the histotripsy. The hypothesis of this experiment was that histotripsy can non-invasively remove luminal biofilms. Here you can see a gross view of crystal violet stained catheters in Figure A showing the control and Figure B showing the histotripsy treated. The crystal violet stain was further solubilized and spectrophotometry data collected at 590 nanometers. Here we can see the histotripsy reduce the spectrophotometry signal to the background level. Finally, here you can see a model of our bactericidal experiments where we treated suspended Pseudomonas aeruginosa and determined colony-forming units per milliliter via serial dilution and plate cultures. The hypothesis of this experiment with this histotripsy will kill Pseudomonas 1,4 in a dose-dependent manner, and the results showed that histotripsy killed Pseudomonas 1,4 in a dose-dependent manner. After 6 scans across the lumen, histotripsy showed an approximate 4-log reduction in viable bacteria. In this graph, you'll note that initial is the number of colony-forming units per milliliter after bacterial culture, untreated are the number of bacteria in control catheters, and histotripsy are the number of colony-forming units per milliliter after histotripsy treatment. Conclusions of these experiments are that histotripsy can penetrate materials commonly used in urinary catheters. As well, histotripsy can remove a bacterial biofilm in catheter lumens. As well, histotripsy has the potential to kill suspended bacteria. Our lab continues to study histotripsy in order to elucidate its uses in clinically relevant biofilms, other catheter-based medical devices, as well as particle-mediated histotripsy methods for targeted biofilm ablation. Lastly, I'd like to acknowledge all those that have helped me along with this project, as well as the Vlasavovich Research Laboratory and all of our funding sources.